Hello and welcome to another episode of Katie the Science Lady. Today's topic is Introduction to Genetics. So let's learn together. All right, so today we're going to do our Introduction to Genetics. This is for those of you that have never tried genetics, never tried genetics problems, never really learned much about them. So we're going to start from the beginning. And today's going to be mostly vocabulary. A lot of genetics, the basics that you need are going to be these vocabulary terms because they'll tell you how to kind of operate these problems and how to think about traits and alleles and all that good stuff that we're going to get into. So we're going to be talking about a lot of key terms today. Genetics, just in case you're like, what is genetics? It's the study of heredity and the variation of inherited characteristics. What this means is it's the study of how we get traits passed on through generations. So how we have traits that are passed on from our parents and then from their grandparents and so on. And this isn't just for humans, this is applicable to plants, animals, pretty much all living things. So we're looking at the variation as well. So the differences in these inherited characteristics in a species. Heredity is just that passing of characteristics from parents to offspring. Again, doesn't have to be humans. You have parental plants. They're the parent generation and you will have offspring. That's what we're going to be calling the babies, our offspring. So that's our genetics version. Because you don't really have baby plants, you have offspring. So it'd be weird if I was calling all these flowers down here baby flowers. So that's what we're looking at here. DNA, we've gone over this in previous videos, but it's the genetic material that our chromosomes are made from. DNA holds all of our genes. So our genes are separate units of DNA that code for a trait. So it's going to be a section of DNA. Each chromosome will have many genes in it typically, and that's what you're looking at are those specific, basically just a section of DNA, those A's, T's, C's, and G's, that's going to code for a specific trait and proteins. For example, in this picture here, brown eyes would be a specific trait, and then freckles would also be a specific trait. And then we have an example over here, the MC1R gene, it's going to code for your freckles. Traits are physical characteristics. Traits, they don't have to be visual. They could be inside your body as well. So they could be internal characteristics. Like some people are able to digest dairy better. They have an enzyme that helps them digest lactose. Some people don't. That's a trait, even though you can't see it. There's also traits like I have freckles. My eyes are hazel colored. That's another physical characteristic. My hair is reddish. So it's another physical characteristic that you could look at. Alleles are one possible version of one specific gene. So we have a trait. Let's say we have a trait for freckles or no freckles. So you're either going to have freckles or you're not. You don't really have an in-between. And so the alleles would be one person could have freckles. So there'd be the freckle allele, and then there'd be the no freckle allele. So there are two possibilities in that case. For eye color, you might have multiple alleles. Or like there are for blood type, multiple alleles. But they're just versions of a trait, essentially. And those are represented by letters. And usually you're going to have capital and lowercase letters, and I'll explain why later. But in this case, big F or capital F would be for freckles, and little f would be for no freckles. And we're going to use that example quite a bit today. A dominant allele is the type of allele that would hide the effect of others. So if something is dominant, it really takes over whatever space it's in. And that's what we're talking about with this allele. So if you have the dominant allele in your genetic makeup, so in your genes, if you have a dominant allele for a trait, it's going to show through no matter what. Those are represented by capital letters, like big B for brown eyes. Brown eyes are dominant. It's part of the reason why you see a lot of brown-eyed people around. But if they have one of those alleles, one big B, inherited from either mom or dad, they will have brown eyes. Recessive alleles are another type of allele, and those will get hidden by a dominant allele. So if something's recessive, it kind of hides in the background. So if you have a dominant and a recessive allele, the dominant one is going to show through. It'll be hiding the other allele. They're represented by lowercase letters, so recessive are going to be like little b. And little b could represent blue eyes. Blue eyes is a recessive trait. If somebody has a parent with brown eyes and a parent with blue eyes, they're typically not going to have blue eyes. It'd be difficult for them to have that. Typically, that brown eye allele is going to get inherited from whichever parent, and it will show through. But we will show you when this might not be the case as well. So again, we've got big B here for brown eyes and little b here for blue eyes. They're both eye color traits. They're both alleles for eye color, but they're different, and one is going to be more dominant over the other, or will show through more often. Okay. Phenotype is the physical appearance of a trait, so it's what you actually can see. The way I remember that is pheno starts with PH, like physical. So it's what you're going to physically see. So hair color, the actual hair color, so red or brown, eye color, green, blue, brown, those are going to be phenotypes. So the words that we use more commonly are the phenotypes. You wouldn't say somebody has big B, little b eyes. That sounds like you're a crazy person. You would just say that they have brown eyes because that's what we would say. It's described with words, so these are pretty common for us. If you would say that's a purple flower or a white flower, or that's a white chicken or a brown chicken, that's what we're looking at here. Genotype, on the other hand, is our genetic makeup. So these are the alleles themselves. So those big B, little b, all those combinations of letters. And you'll see that here. Big P, big P, big P, little p, little p, little p. It sounds funny to say it like that, but that's honestly the fastest way to say it. It takes too much time to say capital or lowercase. It's just easier to say big or little. So you'll hear me saying that throughout pretty much all of my videos. And these are also represented by words, but we've got some brand new language here. We have homozygous dominant. Homo means same. So that's going to be the same case. So either both uppercase or both lowercase for homozygous. If it's homozygous dominant, it's going to be same, homo, same case, dominant. That's big P, big P. And we'll see that over here. If we have homozygous recessive, it's going to be the same. Homo is the same, so same case, but recessive. So that's going to be our little p over here with our white flowers. If it's heterozygous, hetero means different, so you're going to have one of each case. So again, hetero, different, one of each. So one uppercase and one lowercase. So that's going to be our big P, little p right in the middle here. This is really important. This entire slide is one of the biggest things to make sure that you internalize and try and understand as best you can, because when you get to these practice genetics problems, you have to have these down. If you're trying to go back and think, wait, let me think, what is a phenotype? What is a genotype? It's going to take you a long time to answer one of the questions. So let's move on. We already talked a little bit about homozygous and heterozygous, but here are your official definitions for these. Homozygous, remember, means same case or same letters. And that's when the two alleles of the same type are present. It can be any of the two alleles, but they have to be the same. So if it's for brown eyes and you have a big B, it'd be big B, big B. If someone's homozygous for blue eyes, they would have little b, because little b was for blue eyes in our example. As long as they have the same exact type, you're good. Same thing here. We've got our big B and our little b's. Heterozygous is for our different alleles, so you're going to have one of each allele. I think that when you have examples for heterozygous, it's super easy because you just collect all the alleles and you put them together because you know you have to have one of each. And that's what we're going to see here. So big B, remember brown eyes was dominant, so you're going to see that here. Brown is going to show through. Little b, little b was blue-eyed, so that person has blue eyes. Now down here we've got one of each. We have one big B allele for for brown eyes, and one little b allele for blue eyes. So which shows through? Our dominant allele is going to cover up the recessive. So our big B allele, whatever it stands for, shows through. And in this case, it's going to be brown eyes. So you don't mix them. Big B, because it's dominant, will show through. So if you have two parents, let's say you have two parents here, one has big B, brown eyes. One has little b, blue eyes. This is like dad and mom here. If they have a baby, when we do this cross, the babies will end up being big B, little b. So they will all have brown eyes. And I'll show you a good way to visualize this. It's called a Punnett square. And we'll go through a practice problem as well. We call these monohybrid crosses. They are crosses between parents for a single trait. So you can think of it as single trait crosses. We're only going to use one thing. So we'll just do hair color, or we'll just do eye color, or just freckles, but you're not going to combine them in this one, just single trait. That's what mono means, single. So a Punnett square is a method that helps us predict these genetic outcomes. It's a little bit easier than trying to do it all in your head. I am not a mental math person. I like to write things down. I like to kind of work things out on paper. So this is really helpful for me. And most of your instructors or teachers are going to require you to write out your work as well. A monohybrid cross, like we said, that's for a single trait. We will get into dihybrid crosses later, and that's cross for 2 traits. And I'll show you some shortcuts for those as well. So we're going to go with our example here. We've got 2 little birds. They both have green feathers, and green feathers are dominant to blue feathers. So we're going to see what they possibly could have for their offspring, what color feathers their babies could have if they mated. We're going to say that they are both heterozygous for feathers. So they're going to be both big G, little g. Remember, green is dominant to blue, and that's why they both have green feathers. You won't see them with blue feathers because the green is going to show over the blue. So they have both, but green's what you'll see. Here's our Punnett square. It's just 4 squares all stuck together. It's kind of nice. It's easy to draw. No art skills needed. I am not an artist, so this is kind of the limit of my biology art. What we're going to do first, we're going to put one parent's alleles across the top of the Punnett square and the other parent's alleles across the side of the Punnett square. We have to keep them together. So this is one parent here, and over here, this is the second parent. You can't just take both big G's and put them on one side and both little G's and put them on the other side. That's completely different. You need to keep the parents' alleles together on the same side. So again, we took the big G, little g from this parent, put it on top here. We took the big G, little g from this parent, put it on the side here. When you cross a Punnett square, the letters on the top of the Punnett square move down into each of these squares below it. The ones on the side of the Punnett square move across to each of the squares directly next to it. They're not going diagonally, they're not going all crazy all around. They move in just those directions. So this square here, this big G comes down, this big G comes across. Our next square, and that's going to be dominant, so we've got a green baby there. We've got a little g that comes down here, a big G that comes across here. What color should this baby be? It's heterozygous, just like the parents, so it's also going to be green. Same thing with our third square. Big G comes down, little g comes across. Again, big G, little g, heterozygous, going to be green. And then our last one down here, little g comes from the top, little g comes from the side. So this baby isn't going to be green at all. It's going to be blue. So you can have 2 parents that are, let's say, heterozygous for the trait or what have you, and they can have a blue baby in this case. So just because both of their feathers are green doesn't mean their babies will have green feathers. This is what happens if you look slightly different than your parents. Neither of my parents have red hair, for example. My mom has dark brown hair and my dad has black hair, but I was born with fire engine red hair. And this is because it's a recessive trait. Red hair is recessive, so it was hiding behind each of their hair colors. And if I go far back enough into my family history, I have grandparents or great-grandparents on both sides that had red hair. So that's where it gets hidden, and that's why the recessive trait can come out occasionally. If you need more examples, please watch my How-To Genetics video on monohybrid crosses. I will have a whole How-To Genetics playlist for you. There's lots of handwritten examples. I go through this in a lot of depth, so it may be more helpful than trying to rewatch this example again. You'll see me writing things out. I give more tips and tricks as I go through those examples. Thanks for watching. It's really important to understand some of these vocabulary terms before you start tackling some more complicated genetics problems. And if you want to go through step-by-step any of these genetics questions with me for monohybrid, dihybrid, non-Mendelian, including incomplete dominance, codominance, blood type, and sex-linked traits, please go ahead and click on the playlist that should appear somewhere floating beside my head. It's my How-To Genetics section, and it's going to show you step-by-step how to work through these problems with some of my tips and tricks that I've learned over many years of teaching biology. And as always, I hope you had fun, I hope you learned something, and I'll see you later.